So when looking at our genes, we can look at them in one of two ways. We can look at the genotypes, which is what are the actual alleles in our organism, and then we look at the phenotype, or the physical manifestation of the instructions. So if you've ever heard of like big B, big T, little t, if we say big B, little b, then those B's are the genotype. And then if big B, little b stands for brown hair, then the phenotype is brown. And if you have like little b, and that codes for blonde hair, then blonde is the phenotype and genotype is the genes. And sometimes it's easy to get those mixed up. So when we talk about these genes and we talk about how they work in order to make these proteins, we go through this process of basically, it's like reading a recipe here in the nucleus of the cell, and then we have what's called transcription. So if you think about what a transcript is, like you guys will get a transcript of all of your grades from your time here at Akron U. A transcript is basically this copy of all of your grades that gets sent out. So here we have a copy of all this DNA that gets sent out of the nucleus, or like copying a cookie recipe onto an index card, or if you think more along the lines of what we would do these days, if you think about like taking a screenshot or saving something to your Pinterest account. I'm going to have to come up with a whole genes as Pinterest type thing in all of my spare time. So we copy this information to this messenger. That messenger RNA sends it out of the nuclear pores and out into the cytoplasm. So we send our recipe off out of the nucleus. And then it goes through translation. If you think about what it means to translate a foreign language, it's where you basically take the language and encode it into a different language. So taking these genes and lining them up and sticking the amino acids to them and then sending them out as a fully formed protein. So think about it in these steps. We go from recipe, make a copy, send the copy out of the cell, combine all of the stuff together and make what it is that you wanted to make. So it goes through those steps of transcription and translation. So what I just said there, transcription is where the copy of the gene's base sequence is made, and translation is where the copy is used to actually direct this production of a protein. So those are the two main steps in getting from DNA to the final product, transcription and translation. And so in building organisms, and what they are showing here, that it's not actually easy to see, but they have a copy of what's called their karyotype, and a karyotype is a copy of all of your chromosomes. So in order to build each one of these people, as you can imagine, the recipe is mostly the same because everybody's got one nose, two eyes, two ears. So all of that stuff is the same. They have two arms, two legs. They have all of the things that make them human. And then part of their DNA is just different enough that makes each one of these people look very different because they have the characteristics that are passed on from parent to offspring. So the DNA inside of every single one of their cells tells them to be mostly human and then mostly like their parents. And so here you can see, I'm not going to hold you responsible for this at any point in time because this is a little bit complex. But basically during transcription is where they're breaking the DNA apart, unwinding the ladder, and they're taking and transcribing. So taking what's called RNA polymerase, it's an enzyme that helps to allow this mRNA molecule to bind here. And then it stops reading it at a certain point, so it only reads part of the DNA, and then it goes along and it caps and edits it, so it packages it and gets ready to just build the protein that it's going to build. and then it gets its amino acids and, this is the mRNA. This is what heads out of those nuclear pores out into the cytoplasm of the cell, and is ready to get that code and make it into the proteins. So the process of what's called transfer RNA or tRNA is where we transfer those 3 bases, the A's, and we then transfer it from A's to U's, because in mRNA we don't have the thymine anymore. We have what's called uracil, and this is where we put the 3 of them together and we make this amino acid. And the amino acids are put together in a chain until they bind up and make a protein. So 3 base sequences match up, and it comes along, it attaches, makes a little amino acid, and the amino acids make a big long chain of proteins. So our genetic code is really fascinating because we only have something like 20 different amino acids in our bodies that are going to code for proteins. So if we look at all of the possibilities of combinations of A's and U's and G's and C's, we only come up with 20 different possible amino acids. And some of these, you may look at them like arginine is part of like your vitamins, or you think about some of these are found in the foods that we eat. These are our essential amino acids that make up the proteins that make up our bodies. And so we've got our 3, it's called a triplet. We've got the first position is the first letter that's here along the edge, and then the second position goes across here, and our third position is the one that adds the final A's and U's and C's and G's. And so we have 20 different amino acids that possibly can make up our body. So genes are not necessarily always turned on. Sometimes cells turn off genes or turn them on for specific reasons. And that's called gene expression and gene regulation. So this is a really fascinating science because at some point we may figure out how to turn on the genes that make people skinny so we don't have an obesity problem. We may cure several diseases this way. But cells can turn genes on and turn genes off. The way that we look at these are through a process that's called a microarray. This is a scientific process where we monitor the expression level of thousands of genes together. And this is how scientists are figuring out which genes are giving us illnesses, which genes we might go through and mess with a little bit in order to treat the illness, or that we might eventually figure out this fountain of youth that will help people to not age. Might happen. So this goes back to the idea of transcription, that part of the gene process where the protein is binding to the specific sites. We have what's called positive control and what's called a negative control. And this has to do with whether the gene will be expressed or the gene will not be expressed.